See our attendee numbers climbing, so that's good. Okay. Just going to give it a few more seconds, and then I will go ahead and just go over some house rules for this webinar for today. I know we had a good amount of people RSVP, so I don't want anyone to miss out. Okay. Well, I'm going to go ahead and get started. Good afternoon, everyone. Welcome to the Mesulam Center seminar series. I would like to go over some brief housekeeping rules for today's seminar. We will have a Q&A at the end of the seminar lecture, so please feel free to type your questions into the Q&A box in your toolbar at any time during the seminar, and they will be answered during the designated Q&A time at the end. You may also use the raise your hand feature during the Q&A, and then I will be able to move you over from an attendee so that you can join the group of panelists and ask your question verbally. And with that, we will go ahead and get started. I'm going to turn it over to Dr. Rogalski.
Hello, everybody. It's with great pleasure that I get to introduce Dr. Jonathan Rohr today. He's the principal investigator at the Dementia Research Center in Queen Square UCL Institute of Neurology, as well as a consultant neurologist at the National Hospital for Neurology and Neurosurgery. He started working on the neuroimaging of genetic FTD in the early 2000s. He launched and still leads the Genetic FTD Initiative, also known as GenFI, that we're going to hear more about today, which is an international multi-center cohort study of pre-symptomatic genetic FTD. The research of his team focuses on the development of novel biomarkers in frontotemporal dementia, and he runs the annual scientific meeting of UK researchers who are in the FTD field, and support groups through the Rare Dementia Support International. He also leads the FTD Prevention Initiative, also known as FPI, and is the vice chair of the Alzheimer's Association Professional Interest Area in FTD and co-chair for the Enigma FTD group. We wish that you were here with us in person, but are excited to have you with us today, at least virtually, and really looking forward to hearing more about GenFI. So the floor is yours.
Great, thank you very much, Emily. So thank you very much for asking me to attend. I'm in a very early evening, cold and rainy London. And in the modern world that we live, I'm hoping we won't be interrupted, but I do have 4-year-old triplets who've just got in from school. So hopefully if you hear any noise, it's from them along the way. So I've been working in FTD research, as Emily says, for nearly 20 years. And I was just looking back, and it seems like a long time ago, but the only time I've managed to visit your center was during what was called at the time ICAD, is now the AIC, which was in Chicago in 2008. And Emily very kindly showed me around at the time, and I have very fond memories of being there and being in Chicago at the time. So I started research working for Martin Rohrer, who really started a lot of dementia research in the UK. He started familial dementia research and worked with John Hardy in the discovery of the APP mutation in familial Alzheimer's disease. And I was very lucky to start a PhD with him in the early 2000s, and really he taught me a huge amount about familial dementias, and particularly about familial FTD. And at the time, that was really, we only knew about tau mutations, and I look back and see how far we've come, where we're actually coming towards clinical trials now. So I'm going to talk about why we started GenFI and how we've ended up on the pathway to clinical trials along the way. So let me share my screen. Hopefully everybody can see that. So yeah, so that's me, and those are my disclosures. So I don't need to tell you all of this, but I'll quickly flick over. Of course, these are the diseases we're talking about: behavioral variant, and these variants of PPA. And of course, FTD is a complex and heterogeneous disease overlapping with Parkinsonian disorders and ALS. And it can have multiple pathologies. And I like this picture because it just links in the clinical syndromes with all the different pathologies, and just shows what a difficult picture we're trying to solve here. And when somebody who has a symptom sits in front of you, really trying to think not only what their clinical syndrome is, but also what their underlying pathology is and how we might get towards thinking about treating the underlying molecular pathology. So why focus on genetic FTD, which is what I've done for most of my career, although actually I started off really thinking I was going to do a PhD on neurolinguistics, and I did end up doing quite a bit of work on PPA, but progranulin mutations were discovered early on during, well, actually it was sort of towards the end of my PhD, and I ended up falling into doing work really in progranulin and then later C9. But of course, as you all know, somewhere about a third to a half of FTD is genetic. It, of course, allows you to accurately define molecular pathology during life, and it gives you this window into early disease by following at-risk family members. So it's easier to go in this picture from left to right than it is to go from right to left and work out what the underlying pathology is. So what are the genes involved in FTD? Well, actually, there's this sort of rather complex number of genes that have been associated with FTD syndromes. And tau was discovered all the way back in 1998, and then we had a long wait until some more genes came after that. And to sort of make sense of this, I sometimes classify them into three groups. So this is undoubtedly my grouping of them. So I sort of group three. These are genes which have been associated in maybe one or two families. We're not completely clear that they're definitely FTD genes in a sort of Mendelian pathogenic way. And then there's a sort of group two set of genes which are definitely causative of FTD, but are pretty rare. They account for less than 1% of all FTD. And there are some important things. Perhaps the most common of this group two is TBK1, which is an interesting gene. Mutations in it can cause actually lots of different syndromes, behavioral variant or PPA or CBS or ALS, one of the few genes that can do that. But nonetheless, these are all very rare, and most people will only ever see one or two families with these mutations. So group one is really the big three. These are the things that we've been focusing on for the last few years: tau, progranulin, and C9orf72. So if we look at these, I think the epidemiology of FTD isn't great. We don't, I think we're not clear in different countries what the different prevalence is. There have only been 3 or 4 good studies, and most of them are focused on a very specific age group. And often when you read about it, people talk about within the 45 to 65 age group. But actually, there's a really good study done from the Cambridge FTD group a few years ago, looking at a very focused area of England where actually people don't move very often. And they quoted the prevalence across all ages of about 11 per 100,000. So we can sort of then extrapolate that if maybe 30% of FTD is caused by these 3 genes, then the prevalence of genetic FTD, or at least of these 3 genes, is about 3 per 100,000. And when we did a big study of that worldwide, this was the prevalence of the different mutations. So C9orf72 is the most common genetic form, accounts for about 45% out of these 3. Then progranulin, with tau being the least common, and we can extrapolate about not just to what the prevalence of these are, but also about to then how many people are currently living with those conditions across Europe and North America. I mean, I suppose the bottom line is that these are individually rare diseases, and when we come to start stratifying FTD downwards, we're really starting to talk about quite rare illnesses, and that becomes important when we start to think about how we study them and how we might do clinical trials for them. So this was again from our paper in the Lancet Neurology earlier last year, where we looked, we weren't really focused on geographical distribution, but that sort of fell out of the study. And in the US, by and large, C9orf72 is the most common genetic mutation, but there are some important differences in Europe, and in particular in Italy and Portugal and Spain. So in Southern Europe, progranulin is the most common form of genetic FTD. And then in Central Europe, there seems to be an overrepresentation of people with tau mutations, the blue color here. So yellow is C9, this pinky color is progranulin, and blue is tau. So there is some geographical variability. And actually, although we didn't involve these studies, we looked into literature, but actually out in Asia, it's relatively uncommon to see genetic forms of FTD, and much of FTD is sporadic, as I'm sure many of you know. So this is another issue, and this was from an early study that we did when I was looking at progranulin mutations. And what you see here is a coronal section of an MRI. It's taken about halfway through the brain. You just see the bottom of their ears here, and it's on a continuous loop. So it's 3 MRI images taken over 2 years at baseline, 1 year follow-up, 2 years. And what you see over that time period is this shrinking of the left temporal lobe, and huge amount of shrinkage over that time. And you see the ventricles increasing, but very little change over that time on the right, and very typical of this asymmetrical pattern that we often see in progranulin mutations. But the most important thing here, of course, is that the first 2 scans here were performed pre-symptomatically, and only at the last scan had this person become symptomatic. And so even on an individual basis, we can see this pre-symptomatic atrophy. So this tells us that we know that the disease process starts many years before symptom onset. So in reality, there's a short clinical disease period and a long pre-symptomatic period. And so we're seeing people in our clinics really when their function is starting to go off. But we know that we can start to detect some subtle social cognition and behavioral problems just before symptom onset. And with some more detailed psychology tests, we can perhaps push that back to 5 to 8 years. With MRI, we think we can perhaps see some change, starting to see some changes maybe 10 or 15 years before. And then maybe we can start pushing that back with functional MRI and PET and CSF. And certainly the evidence we have from Alzheimer's disease and particularly from the familial Alzheimer's disease study, the DIAN study, is that potentially we may be able to see some of these changes in the key proteins 20 or even 30 years before people develop symptoms. So the question was then, how do we end up solving these challenges? These are rare diseases. There's geographical variability in the prevalence, and then these are diseases which start before we develop symptoms. So we'd studied families for many years, but we realized that really the way to do this was to bring specialist centers together across distributed geographical regions to try and actually provide a uniform platform for how we would study it. So GEMFI, which I'll talk about mainly, was the first study to do this, but we've been followed up by All FTD, which is the huge US study. Initially 2 studies, Artful and Lefties, now come together. There's also an Australasian study, very similar now, DINAD. And actually, as many of you who will have been to the ISFTD last week will have heard, there's also REDLAT, which is a South American study, very similarly. And as you'll hear, we're sort of coming together to think about how we all work together. So GEMFI was really set up because we realized we couldn't do it on our own, and we realized we had to set up a common way of us all studying genetic FTD. And we started off actually nearly 10 years ago now with 5 centers, grew to 13 by the end of the first phase, to 26 in the second phase, and we're just about to enter a third phase where we'll be more than 30 centers. And we focused very much on pre-symptomatic periods, but we also do have people who are symptomatic in the study. And again, we focused early on the big 3, but we have also, in the second phase, we added some people with some of these rarer group 2 mutations as well. And I think the key to the success of GEMFI has absolutely been about standardizing assessments. So here are all the sites that are involved in GEMFI. the reason we involved Canada was just how we ended up getting the first grant. The first grant happened to be a European and Canadian grant call, which is how we ended up pulling the Canadians into our European study. But now we've eventually slowly gathered together really all of the big FTD centers in Europe into the study. And so we're really now right the way up. Our most northern center is in Oulu in Finland, right down to our most southern center in Coimbra in Portugal. And we've been very lucky, really, in terms of recruitment. We've sort of had a slow increase up. We have these data freezes where we release data for people to look at. And we're just coming now to the end of this GenFi 2 period. And this has been, we still have managed to see some new people during COVID, although we had a period where we weren't seeing people, but actually some centers in Europe where it was slightly better, COVID was better controlled, did actually start to see some people, particularly in the middle of the year. So for those interested, and all our publications are available, this is the website, genfi.org. And so I think what I'll talk through is hopefully some of our highlights. So certainly we're nearly 1,200 people in, and we are have quite a number of publications now, some of which I'll talk about, and we're just about to enter this third phase. So what I'll talk about really for the rest of the time, and then before I talk briefly about clinical trials, is really what we've learned over time. So I'll talk a bit about demographics, a bit about clinical and cognitive measures, and then go on to some biomarkers, talking about imaging and fluid biomarkers. So talking about demographics first. So this is from this paper by Katrina Moore, who was one of our PhD students. It's very clear, and anyone who studies this will know that individual age at onset is very variable. And you see that here on the left. This is the variability in symptom onset in progranulin, in onset and death, progranulin, then tau, then C9. You can see there's a slightly younger age onset in the tau, very similar in the C9 and the progranulin. But there are clearly people who are still becoming symptomatic in their 80s and even into their 90s with progranulin and C9. So this is a hugely variable age at onset. And that age at onset is very poorly predicted by the age at onset in other family members. So either by the parental age at onset or by the mean age at onset in families. It's best predicted in the tau mutations where actually the correlation is not too bad. You get an R of 0.63 when you look at the correlation of individual age at onset with a mean familial age at onset. So best predicted in tau mutations, then C9, and then very poorly predicted an R of only 0.18 and 0.22 for progranulin mutations. And people know that if you follow progranulin mutations, we certainly see siblings who might have 10 or 15 years between their ages at onset. So thinking about the clinical and cognitive side of things, we've had lots of studies within GenFi looking at symptoms. I think one of the nice things about GenFi is really that virtually all of the centers have been very keen to contribute towards the analysis of doing things. So these are 3 studies by the Canadian group of Liz Finger in Ontario, by Barbara Baroni's group in Brescia, and by James Rowe's group in Cambridge in the UK. It's clear that the symptoms vary between the different diseases. This is cross-sectional variation in symptoms. And here, there's a nice paper just come out from Barbara's group showing the change over time in the different behavioral symptoms that characterize the Roskovsky criteria, and how those vary and how they vary between the different groups. This is a nice paper that's just come out showing actually, if you look at apathy in particular, it predicts those people who will decline. So if you look at particularly the pre-symptomatic period, this will then predict who is likely to decline in executive function later on. Of course, in clinical trials, we're particularly interested in thinking about how we might use different clinical rating scales. And we learned a lot from the study of the ALL-FTD study, who've studied this extension to the CDR, which we were calling the FTLD CDR, now calling the CDR plus NAC FTLD. But we have 2 scales which have been fairly well validated. This FTLD CDR and this FTD rating scale, which actually here you can see the correlation between them. They're fairly well correlated, R of 0.77, although there are some differences. And here, these people are stratified by their baseline global CDR score. You can see that there is some measurable decline in a 0.5, which might be a prodromal age, but particularly as soon as you become symptomatic, over 1 year, you start to see this decline, but no real decline when you start off at a asymptomatic period. So the FRS and the FTLD-CDR are hopefully going to be helpful, at least in the symptomatic period, potentially in the prodromal period, but not really in the asymptomatic period in trials. I'm not going to go too much into this, but just the headline here is certainly cognitive impairment is obviously different between the different genetic groups. And of course, we do see cognition becoming abnormal pre-symptomatically in some studies. So this is just 2 studies. This is a semantic knowledge test, a modified version of the camel and cactus, where we saw, if we looked at early and late pre-symptomatic, we saw changes in the late pre-symptomatic period, in tau mutations and C9, and that probably goes along with, particularly for the tau, some early semantic change. And here, this is in the Ekman faces, and here you see really only early pre-symptomatic change within the C9 group. And we're just going through all of the cognitive battery at the moment and looking at that, and it's very clear there are differences, more memory and semantic knowledge changes in tau mutations, and then more widespread cognitive change with some posterior change in the C9 group. And we've thought about how would we combine these. Maybe we could bring together a composite score that would help lower sample sizes. And this is a study just we're finishing off, which was a collaboration with John van Sweeten's group in Rotterdam. And here you look at the coefficients that are being shown. This is the GEMFI battery, language tests, attention, executive function, visio-construction, memory, and social cognition. And you see that things that end up in a cognitive composite are different between them. In tau, as I said, all the memory tests end up in a tau battery along with the camel and cactus, so a semantic knowledge task. But we see different things across the tau. Only in C9 do we see the more posterior sort of visio-construction tasks. And when you look at using these weighted or average composites, the numbers end up being substantially smaller. This is with an effect size of 0.1 or 0.2. You end up needing substantially less people than you would do if you used any of these tests individually as a cognitive outcome measure. So thinking about MR imaging, these are some studies from at the start of GEMFI. This was the first study we did in Lancet Neurology in 2015, where we looked at estimating onset. And what we saw were there were different patterns of atrophy, and that the atrophy was different in different areas before onset. So in tau mutations, you see very early change in the medial temporal lobes and the hippocampus and amygdala. In progranulin, it's in the insula, and then in the temporal and parietal lobe. And those are sort of 10 to 15 years before estimated onset. Whereas in C9, you see this really early onset, maybe 20 or more years before onset, particularly in the thalamus, which we've come to understand is important, but also in more posterior cortical regions. And this is a different study where we just looked at symptomatic carriers and looked at the different patterns of atrophy. The yellow is the areas of atrophy that overlap. And that's interesting because that's in the orbitofrontal lobes, it's in the cingulate and in the insula. But then there are different things between the different mutations. So anterior temporal lobes for tau, more superior frontal and striatum for C9, and then more thalamus, cerebellum, and posterior cortical for the C9, the striatum and the superior frontal for the progranulin. So different patterns of atrophy for the different groups. And important to recognize, it's clear from that VBM study, that atrophy isn't just frontotemporal, and there is more posterior cortical and subcortical involvement. And this is a nice study from Martina Bocchetto, who works in my group, and she has done regions of interest study where she's looked at individual subnuclei and subregions within all of the subcortical structures. And interesting, if you look at this according to the baseline FTLD-CDR, when you start to look at these individual subnuclei, you start to see changes even much earlier than you see when you look at the whole structure. So here, The red is changes during the completely symptomatic period, a CDR of 0. And you see changes in C9 in the amygdala, in parts of the hippocampus, in the thalamus, parts of the hypothalamus, parts of the cerebellum, and also in the cortex. And that's sort of described in some ways by other people. But here, this is interesting. When you look at specific parts of the hippocampus and amygdala and specific subfields and subregions, you start to see changes in the completely asymptomatic periods and not just in the prodromal periods. Progranulin is a disease, I think, where you see relatively late change. And here you see a change only in specific regions in the 0.5 prodromal area. And here's a nice study, again, from Liz Finger's group in London, Ontario, where actually, of course, it's not just frontotemporal and subcortical regions, but actually the ventricles do expand over time. And you see the ventricles expanding differently than controls about 5 years before onset. We've started to think about how we do more sophisticated analyses. And here, this is using a new machine learning analysis called SUSTAIN. The first paper was published in Nature Communications a couple of years ago. And here, you just look at the imaging and see whether the data clusters. And actually, it clusters very nicely, but interestingly, not into 3 groups, but into 4 groups. And so there seems to be a progranulin group, very asymmetrical, a tau group, very focal temporal at the start, but there seems to be 2 C9 groups, one a bit more subcortical and one starting a bit more frontotemporal. And interestingly, as we've moved forward and we've got more people in a tau group, we realize you can also split these into 2 groups. And actually, this replicates a paper from many years ago by Jenny Whitwell, where she looked at the difference between certain mutations. And here, if you have these intronic mutations or mutations close to the intron, you get this more focal temporal involvement. But actually, if you have the P301L mutation and mutations further away, you get a much more frontotemporal and anterior cingulate involvement. So even within tau, you can pick out different patterns depending on the mutation. We don't understand yet how these split or what these 2 groups represent. We've also looked at things that might modulate brain volume, and particularly Barbara Baroni's group has looked at how education modulates brain volume. We've also looked at different things, not just T1 imaging, and we know that in progranulin, they rather have these unique white matter hyperintensities. Here, a very nice picture of where those white matter hyperintensities are. That's the red area. You can see they sit in between these fibers and disrupting, really causing a connectivity issue by sitting in the middle of these white matter fibers. And we see that in this diffusion tensor imaging, which does show as pre-symptomatic white matter changes, but that occurs in all diseases, not just progranulin, but also C9, where you see very early pre-symptomatic change. We can also see very early perfusion change. And here's a lovely study from the Toronto group, Mario Marcellus, where you see very early perfusion changes with ASL. fMRI is a bit more interesting. These are 2 papers from James Rowe's group in Cambridge. They seem to show that functional connectivity actually is rather maintained until symptom onset. Here it suddenly drops as you head towards symptom onset. And their theory being that actually there's something that maintains the functional connectivity even in the presence of the structural abnormalities. And here, this is a very recent paper from one of his group showing that helps to maintain cognition nearer to onset than you might expect given the patterns of structural loss. So lastly, we'll talk a bit about fluid biomarkers. We've got 2 great biomarkers which are going to be super helpful for trials. Progranulin we can measure in CSF and we can measure it in blood, and low progranulin levels are, of course, essentially diagnostic for a progranulin mutation and are low both in asymptomatic and symptomatic carriers. We also have this nice measure now, the poly-GP dipeptide repeats, which is a direct marker of those toxic dipeptide repeats in C9 and are raised in both pre-symptomatic and symptomatic carriers. And I expect these are going to help to allow a direct measure of a therapeutic effect on pathology in trials. But we also have this nice measure, neurofilament light chain, which is just a very generic measure of neurodegeneration. It's abnormal in lots of diseases, but it's clearly abnormal in particularly the symptomatic period. Here's an initial cross-sectional study. Here's a longitudinal study published last year, or 2 years ago now, in Lancet Neurology. There's a slow increase in pre-symptomatic carriers, and then this sudden increase during conversion. and then it sort of slows again during the symptomatic period. So this sudden increase seems to be indicative that you're about to convert. And this is a paper in Press where we combined all FTD data, which is along the top, and GEMFI data along the bottom. If you have a cutoff level for NFL, you can predict who, both from an asymptomatic period and from a CDR of 0.5, is likely to progress to symptomatic over the next couple of years. So NFL helps us also predict who's going to convert to symptom onset. We're also interested in other areas. We know neuroinflammation is important in progranulin, and we now have this nice blood measure, GFAP, which is abnormal in symptomatic but not pre-symptomatic people with progranulin. And we're also interested in looking at things like synaptic dysfunction. And we have this nice measure now, neuronal pentraxin 2, which again is abnormal really mainly in the symptomatic period. So I'll lastly talk about this connection of all of the genetic FTD cohorts across the world, although it really started between the US and Europe and Canada, the FPI. And it's mainly led by myself and Adam Boxer, but also involving Brad Beauvais and Howie Rosen, and these are our coordinators. And we've really trying to bring together all of these studies across the world, but also involving patient support groups and different advocacy groups and charities as well to bring together. And here again, if you're interested, here's our website to think about that. Our key goal is really to try and bring all the studies together and work with pharma for us to properly design trials and to make sure we have uniform standards for trials, make sure we have an international database for trials, and that we can get people to share things. Really, the first people off the block and the people furthest forward are this company, Alector, who have an anti-sortilin therapy. Sortilin tags progranulin for breakdown. If you block sortilin, then you have more progranulin available. It's no longer broken down. And this is their data from their phase 1 study, that giving that drug can increase progranulin levels back into the normal range. And Alector have just started their phase 3, which we're very excited about and to see. Now, really the big story for the next few years will be these genetic trials. So for toxic gain of function, we're likely to be mostly interested in antisense oligonucleotides, so small pieces of DNA which are going to block the messenger RNA and hopefully essentially silence the gene, often called gene silencing therapies. So in C9, there's already a trial underway by Biogen, mainly in ALS, but hoping to be an FTD, and this other company, Wave, who are starting to get their ASO study underway. Same with tau. Hopefully there'll be a Biogen study. Biogen have a study of this tau ASO in Alzheimer's disease, but haven't started yet in tau mutation carriers. Progranulin is the opposite. There's also a loss of function. And what we're trying to do is increase progranulin. So the aim is to give an AAV, an inactivated adenovirus that has a working version of the gene, and give that back into the body. And there are two companies really focused on that at the moment, Prevail and Passage Bio. And this is really hopefully the future over the next few years. And we've worked very hard thinking through GEMFI and all FTD, how we would think of the outcome measures that we would use in these trials. And we've been able to work with companies to think more about that. One of the most important things we've done is really create a participant engagement board that's mainly been in GEMFI at the moment, to really be a voice of the participants in the study. And we've really involved them both in the design of the observational study, but really linked them in with pharma to thinking about how we design trials and clinical trials, and making sure we really have the voice of the participants involved in that. And here's just one thing we've done, which is one questionnaire. Are people more likely to find out their genetic test results to be in a trial? I think very few people, certainly in our experience, about 20 or 30% of people know their test results. It's a bit higher than our normal number here, but certainly I suspect more people find out their genetic result if they know they might be in a trial. And the problem is lots of these trials are going to be very interventional. Some of them are going to require lumbar punctures, and actually it's interesting, lots of people, up to 70% of people are happy to have one and have regular ones. For the Alector trial, it's interesting that Alector are going to offer the NFL as a way of stratifying people. And we asked people, would you want to know, were you nearing symptom onset? And about 60% of people said they did, 40% of people said they didn't. And then we thought, well, what other things? Maybe would people be happy to have an IV treatment? And actually, the majority of people are happy to have an intravenous therapy. But actually, obviously, interestingly, lots of these therapies might be very interventional. They might go into the back of the neck, they might go into the head, and obviously smaller numbers of people would be willing to have much more interventional studies, but nonetheless, quite a number of people, 2/3 of people still willing to have something injected into their head, about 53% of people willing to have something actually directly injected into the brain. And I think, we will obviously moving to an era that is slightly uncomfortable for everyone, doing therapies which we haven't done before, where we are giving people potentially quite interventional therapies. And certainly most people are happy to be in a placebo study. So, it's amazing. I think that the FTD community is such an amazing community. We're a relatively small community, but I've always been, for the years I've been working in it, it's been such an amazing, friendly community. And really, the key to solving this is obviously worldwide collaboration. It's a rare disease and we need to work together across patients and families and academia and industry. And I think we've really moved forward, but there's still lots of challenges and there's still lots of work to be done in trial design and how we develop biomarkers and we validate it. And really, the goal of creating the FPI, this worldwide group, is really that we can provide the best science and the best opportunities for patients and families that hopefully will eventually lead to a cure for FTD. So there are far too many people to thank, but above all, really the participants and caregivers and family members who take part in the study. We have lots of amazing PIs across the study, but I have an amazing team. We have a website called FTD Talk. It's fairly UK and Europe based, but there's lots of interesting things on there, and we try to blog about our work, and hopefully there might be some things of interest to you on that. So that was a very quick run through our work, but hopefully an idea of where we're heading, what we've done, and how we're really have worked our way up to really being at the stage where, very excitingly, we are, at this precipice of entering lots of people into clinical trials.
Thank you so very much, Dr. Rohr. It was a real tour de force and just so impressive how much you've been able to accomplish in over a relatively short period of time, and that it is through the collaboration of across the world that we know so much more. I remember when I first started in graduate school, there was FTLDU, so that there was, or dementia, DLDH, dementia lacking distinctive histopathology, and to think where we've come from then till now is truly exciting. So thank you for putting it into context and giving us a kind of a timeline too of also where things happened and where we've been and where we're going. So I want to encourage our colleagues to put questions in the chat for Dr. Rohr, and if you would like to pop on and list your question live and in person, let us know, and Lisa can make that magic happen as well. I see Marcel coming on now.
Jonathan, welcome to Chicago.
I really wish I was there.
Well, so do we. So I just want to echo Emily. This was a fantastic lecture. What an unbelievable amount of work. So eventually, when the happy day is going to come and we're going to have effective means, I think what you did is clearly going to be the prime mover. So it's amazing. Now, I have one detailed question for you. Do you have enough cases to be able to look at both behavior and atrophy with modifiers, such as TMEM 106B, for example?
Yeah, the answer to that is probably yes. So we are just getting to the point where we're going to get some of that genetic data and being able to look at it. I mean, I think we've sort of slowly grown over time. it's been a sort of, in some ways, I give you the nice rose-tinted spectacle view of things, but we've obviously struggled to get funding, and we've slowly grown in numbers. But I think we're sort of getting to the stage where we have good enough numbers to start doing some of these stratified things, and I think Team M106B is, of course, the most interesting of the modifiers that we know about. And I think we'd love to go back, and particularly we're doing some work at the moment looking at the difference between resilience and non-resilience, where we're defining kind of resilience as those that are making it into their 70s and 80s without having developed symptoms, versus a sort of non-resilient, those that have somehow managed to get symptoms in their 30s and 40s, and seeing what is the difference between those groups. And I suspect Team M106B is likely to, of course, be one of those things, but hopefully we'll be able to pull some other things out of that. And I think, I don't know how you feel, but I often struggle when I talk to people about their risk, and when I see family members and I say to them, well, look, your risk is 50% that you will carry this mutation. But that's slightly different than saying your risk is 50% of developing the disease and developing symptoms, because we know that there are going to be a group of people, there is age-related penetrance. We know there's going to be a group of people that are going to live to their 80s and 90s. And I feel sort of, at the moment, fairly helpless in being able to give someone more specific. It'd be nice if we had a way really of being able to say to people, to stratify people better. I mean, that was really the starting point of that Lancet Neurology study, was how do we give people better information when they're sat in front of us in clinic and they say, I've inherited this, but when am I going to get it? And I still find those discussions very, very difficult. I hope Team M106B, when we can do a bit more work on it, will be able to give people a bit of a better answer to it, and maybe be something that we can offer people, and maybe be able to stratify things a bit better.
This reminds me of a patient that I had whose mother had a MAPTA mutation. He was asymptomatic and wanted to do genetics, and he had the mutation. Absolutely devastated. So I was working with Mike Hutton at that time on that patient. And so Mike wrote back in to say this particular mutation is really one of those that penetrance. It allows one to live until pretty ripe old age, and that made all the difference in the world.
Yeah, I think that's right. And I must say, I do find, I don't know if you have the same experience, but even just talking about the age-related penetrance, often people have come to me having maybe met someone else and talked a bit about FTD with someone else. And actually, when we have that conversation and I say, look, actually, there is a lot of I think a lot of people automatically assume they're going to get symptoms the same time maybe as their parent. And actually just even saying to people, look, there is this difference, and there are some people that will live into their 80s and 90s. That does actually make a big difference to people when you have that discussion about the fact that there is that variability. And I think when we think about the very personal basis, and the thing I've enjoyed the most about the research is that personal basis, and I've got to know a huge amount of families. I mean, I talked about working with Martin Rosser for years, and I'm now seeing children and the grandchildren of the people that Martin was seeing, and the families that Martin kind of brought together in the 90s. I know those families now, and you have these very difficult discussions with them, and it's very difficult. I think it's good to be able to not necessarily sugarcoat things, but have a real story, for people to be able to get the real information. And I think that's, people want information from experts, particularly when it's a rare disease, and they want to get that information. I mean, I think that's, you guys have lots of experience of that, and particularly in the work, Emily, you're doing in the PPA group, of really that connection of people getting that expertise.
So Sandy's got a question that I'm going to ask in just a second, but I kind of want to stay on this theme of age of onset and ask a question there. And in our experience here with sporadic PPA, age of onset, of course, it's not the same as it's subjective. And so that you can find that the first time you see someone, they say, oh, it's been 1 or 2 years, and then for certain individuals, they think back, and so the next time we see them, they may come back and say, oh, I remember this time now 7 years ago when I was going to say something and I couldn't think of the word. I think that's when my disease started. I wonder, in your group, how have you navigated that now? Because you have families with a genetic mutation. For the pre-symptomatic and asymptomatic individuals, do you find that they're leaning towards, they're looking for those first signs? So maybe you're inching towards an earlier age of onset in that group in any way?
I think that's right. I mean, I think in the Lancet Neurology paper, we replicated some work from other people from the Belgian group and from the French group, but also extended it to Tau, where we showed that when you look at 2 generations next to each other, the second generation, by and large, have a shifted age at onset. And the Belgian group, the Christine van Brochem group, just did it in C9, and they argued that was anticipation at the time. But then the French group, Isabelle Leber's group, did it in progranulin, and they said, look, it's not anticipation because there's no possible biological mechanism how progranulin could potentially have anticipation. These are the same, they're just loss of function mutations. And we found the same in Tau. And I think at least part of that answer is what you say, which is, and I see it when I see the people, which is they are on the lookout. They're always on the lookout for symptoms. And we sometimes can often accelerate that thought by, number one, them being in a study. We make them focus on their symptoms when they come and visit us once a year. We talk about it. They come to our meetings and they hear in the support group meetings about symptoms. So I think absolutely people are just much more aware of it. It's a real struggle. I think there's probably going to be a couple of papers coming out, and I know Adam and the All FTD group have thought about this, and we've thought about it. It's a bit about how you define prodromal FTD and how you do cutoffs. So what's the cutoff for start of a prodromal period, and what's the cutoff where you convert, and what does that mean to convert from prodromal to fully symptomatic? And I sort of think about that in the Huntington's world, where I look at these studies where they talk about pre-manifest carriers, and then you look at how they define symptomatic, and it's very strict, and actually you're quite symptomatic in Huntington's before you're really defined as symptomatic. So it's an artificial boundary. And in some ways we have to create some artificial boundaries that will help us in research. But it's hard to know what those boundaries are. And I think it's easier when it's a motor symptom. It's substantially harder when it's a cognitive symptom or a behavioral symptom. And we've got a whole working group at the moment thinking about this topic, and I find it endlessly difficult of thinking how do we get back to that. I mean, I think just talking about sporadic PPA, I had a very interesting person with a logopenic variant who was a producer of a sort of science program in the UK, and because of that, she'd been interviewed many times on the TV. And she went back and they brought with them, we looked up together on the internet a few videos. And there's an amazing video of her on the BBC website of her being interviewed 10 years ago. And she makes a number of very subtle pauses in her speech. She makes one phonemic error of a quite difficult word. And it's very clear in retrospect that they had it. And actually the family will now say, because they're quite tuned into it, they say, actually, yeah, we thought about it at the time. I know we've told everyone else it started 5 years ago, but actually now that you tell us, actually it was 10 years ago. And this was, we've looked back at this video and we can spot that there was a problem. So I think it's a super difficult topic. I suspect the solution in a research basis will be providing some arbitrary boundaries that we use that are just helpful to place people into groups in a way we kind of do that now with the FTLD CDR if we're starting to call 0.5 prodromal and 1 fully symptomatic, that's already starting to create some artificial boundaries of how we start to define these things. But I still actually, you know, and drug companies ask us this question: how do you operationalize? It's the operationalizing of the behavioral and the cognitive symptoms that I still think is tough, and varies, of course, by the educational level of the person talking to you as well. So yeah, an endlessly fascinating topic, super hard. Maybe even harder in C9, where there are some of these neuropsychiatric symptoms, but something I think kind of active and something we probably all need to solve as a kind of worldwide group.
Yeah, definitely agreed. So Sandy says, Dr. Weinjob says, this is very impressive work and a real highlight in the linked voyage to treat FTD. How do you think the mutation model applies to so-called sporadic FTD?
Yeah, good question. I think I still do run. So we run a sporadic study at UCL as well. We certainly see people. We've got some amazing people who I work with who are also interested in PPA, and so we still do a lot of work in that. I think it's going to be difficult. I think it is going to be difficult. I think if we can push back early diagnosis, and that's looking at subtle ways of thinking about language symptoms and about behavioral symptoms, we're still not going to get to, it's going to be very hard to understand a pre-symptomatic period in sporadic FTD just by the prevalence of it. And I don't have an easy solution to that. There is one amazing study, which I think is often lost. I think it's by Pardini et al. In the JNMP. I don't know if people know this study. They did the reading the mind and the eyes task, social cognition task, in a general population. And they took the people who scored badly at it, and I think they did it in the thousands of people in a particular area in Italy. And then they took all the people who scored badly at it, and they followed them over time. And they showed that a small percentage of those people ended up developing FTD. I mean, that's a remarkable, it's the only real pre-symptomatic study of sporadic FTD, and amazing to be able to do that. So although it's hard, and I think, you know, when we think about healthy aging studies and how many people are in all the different healthy aging studies around the world, the likelihood of there being people who develop FTD within that are going to be a small number of people. But we still need to think about, are there ways that we're going to be able to pick up those people in some way? And I think that study in some way is reassuring that actually there are potentially some ways that if you do a large screening of people, that maybe you can start to pick up some people who do it. So I think it's not a perfect model. And, you know, I'm afraid to say I've definitely, you know, fallen foul of writing in grants the importance of the model that I present to wider FTD, but it isn't necessarily. And I think it's going to be much harder to solve it and much harder to push back, particularly if we get to a stage where we think that the best way of treating things is going to be, you know, before people develop symptoms. So tough. And you know, it's been easier to get to the point where we think about getting to genetic FTD, but, you know, all these people come to our support groups and come to my clinic and they say, Dr. Rohrer, you know, what about sporadic FTD? And I get the what about sporadic FTD question a lot. And I sort of say, well, you know, we've moved on, but it is going to be difficult.
Thanks. We've got a couple more questions. The next one is, out of curiosity, would any of these therapies, specifically considering the C9orf, be helpful for treatment of ALS as well?
Yeah, definitely. So, well, ALS due to C9. But yeah, so I think one of the things that we've been struggling with is also for C9 specifically, going back to the previous question, is how do we also combine our clinical measures, and particularly prodromally, how do we think about prodromal motor measures and what you might call mild motor impairment? So you're not just thinking about, mild language impairment, mild behavioral impairment. We're also thinking about mild motor impairment for people with C9, because we don't know what they're going to get. We don't know whether someone with pre-symptomatic C9 is going to get ALS or FTD or a mixture of FTD ALS. That makes it very difficult. Now, interestingly, Biogen decided that they would do their trial for the C9 ASO only in ALS to start with. And you can understand the reason for that. It's easy to focus on. They'll have one outcome measure. Wave Life Sciences, however, are going to do both. They're going to think about how they would combine, and they've been talking to us in both All FTD and GenFI about how we might use measures that could potentially combine motor and behavioral and cognitive measures. The eventual decision, of course, will be the FDA. Will the FDA allow a later phase trial where you get a mix of phenotypes within it? The talk at the moment is they won't, which means that someone like Wave or Biogen would have to do 2 trials, one in ALS, one in FTD. And of course, where do the people with FTD ALS overlap fall? Can they go into one study or the other? So it is difficult. And again, it's not something we've completely solved, I think. And I think the work that's been done on the CDR is great, but it isn't the complete answer. And it isn't a complete answer, particularly when the FDA would like to have a clinical and functional defined outcome measure as the primary outcome measure for trials. That makes it tough when you're adding in a motor component onto the issues that we deal with on top of things.
Sure. Well, I think it was exciting to see last Friday when there was the AFTD organized the meeting with the FDA for individuals living with FTD and their family members to have a voice and share their stories.
I thought that was a great meeting. I was sort of dipping in out. So I had, unfortunately, the ISFTD for UK timing was not great for those meetings, but actually it was great to listen in. And we actually had a few GenFI members, participants who gave their testimony, and it was really, lots of the testimonies were so powerful and just amazing, and really great for the FDA to hear those stories from people.
Yeah, incredibly powerful. I see that Dr. Weintraub has popped on here. Sandy, did you have another question you wanted to follow on with?
No, I'm trying to get my video on. Hi, Jonathan. Great to see you.
Hi, Sandy.
So I asked Bruce Miller this question after his talk at the ISFTD about, this model of the precursor, the prodromal. We've gotten that from Alzheimer's disease. And so in Alzheimer's disease, we know that if you live long enough, everybody's going to make that, but not everybody's going to make. So what do you think about Alzheimer's as a model for FTD?
I think it's difficult. It's different disease. Just taking a step back to genetic FTD. I think these diseases are different. The 3 diseases are different diseases. Progranulin, if you look at it, there's almost no neurodegeneration really until nearing symptom onset. And then there's this hot acceleration downwards. And then tau is a bit the most Alzheimer's like. There's probably some slow buildup of tau. You get this slow neurodegeneration, maybe 10, 15 years before. And then it slowly goes down in a very kind of Alzheimer's-like pattern. And then C9's even stranger. It's potentially got this neurodevelopmental issue around it. Brains in the 20s are not quite normal. There are this group of people who are really slowly progressive. So Alzheimer's is not the right model for the majority of FTD. And the question then is, well, what about our sporadic people? What's happening to them before symptom onset? Are they slowly building up over time? Are they not? Are they, have they got neurodevelopmental problems? As you guys have shown very clearly, the neurodevelopmental issues and around learning disability. That's very clear. And then, maybe we can't, I think what genetic FTD teaches us is that we can't extrapolate backwards in time from symptomatic FTD in the same way that you can. And I often show this picture with these sort of actually different trajectories for the 3C9 groups, because they are different. They're definitely progressing pre-symptomatically very differently, and after symptom onset, they're progressing very differently.
Thank you.
Maybe to kind of piggyback on that, we have one more question, and then we'll let you go. So Adam Artistek says, really great talk, Dr. Rohr. I saw on your FTD version of the Jack et al. Abnormal biomarker curves that you placed fMRI with FDG and before structural changes, but later presented that fMRI didn't change till near symptom onset in the Rittman paper. I wonder if you could expand on those two different perspectives.
Yeah, that's good. I will tell you that is an ongoing argument in the group. I still have it in my slide, in the curve slide, because in my heart of hearts, I sort of believe that we will be able to detect functional deficits, and it's how you look at it and the different measures you look at it. James Rohr is a really he's a great collaborator. He's up the road from us in Cambridge. I mean, he really very forcefully says, look, when you look at the measures that I'm interested in, when we look at these very specific graph theory measures, we're not seeing it. We're just not seeing the change. And it is difficult. They've looked, their first study was in a relatively small number in one of our early data freezes. Maybe it's power. But I will tell you that it is one of the things that generates one of the most heated discussions in our group about whether there is, these two, they are essentially two opposing theories, which is surely there must be functional change before structural change, and no, the structure, the functional change is maintained, it's not happening, and actually we're maintaining it to onset. So you very cleverly noticed that ongoing argument, but it's definitely a topic that we're interested in and thinking about.
Well, wonderful. Thank you so much. It's been a real pleasure to have you here remotely, and hope all is well there. And thank you for a really fantastic talk and the amazing work that you're doing.
Amazing. Thank you, Emily, Marcel, Sandy. Thanks to Lisa for all the organization. I will get back to putting my children's bedtime in the UK. So I shall be reading stories about Chicago to them tonight. Thank you all very much.
Very well. Thank you.